Christians oppose science when they claim that God's handiwork is evident in the design of living things?
Or does Christianity open believers' eyes to the beautiful and brilliant design of nature and particularly living things?
The Earth teems with exquisite organisms.
History tells us that many of man's greatest technological achievements are actually reverse-engineered machines that already exist in still more elegant forms in nature.
The complexity of living organisms is matched by the elegant efficiency of their apparent design. Biology is the study of complicated things that give the appearance of having been designed for a purpose.
Has evolution saddled humans with minds that see design where there is actually none?
The eminent Darwinist Richard Dawkins certainly thinks so.
It is almost as if the human brain were specifically designed to misunderstand Darwinism and to find it hard to believe. We are entirely accustomed to the idea that complex elegance is an indicator of premeditated, crafted design. It took a very large leap of the imagination for Darwin and Wallace to see that, contrary to all intuition, there is another way.
Is it rational to believe that living things look designed when they are in fact designed?
The answer to that question depends very much on what one believes unguided nature is capable of accomplishing and what one believes about God.
Let's start by looking at what nature can do all by itself.
Unguided nature has two tools to work with. The first is a set of laws governing how matter and energy behave. The second tool is chance, the apparently random conditions and events that ultimately lead to interaction of matter.
Let's look at living organisms to see whether nature acting alone might explain them or whether they require engineering imposed from outside.
Living organisms are made of cells, which are composed of very specific biological macromolecules. These large molecules include proteins made of amino acids and nucleic acids like DNA, which are made of nucleotides. Molecules are the bedrock of all living things. So if nature cannot make the large, complex molecules found in living things, then even at the simplest and most fundamental level, life requires a designer.
Let's start with proteins. Protein molecules resemble a language in that they consist of amino acids that have been strung together like letters of the protein alphabet to fulfill a meaningful purpose. The potential number of different ways in which amino acids can be combined into a moderately sized protein is far greater than the number of particles in the known universe.
Out of all these potential combinations, only a small proportion of amino acid sequences result in functional proteins that can sustain life. In some cases, slight changes in the amino acid sequences of even a single protein can cause death.
Before the first living thing, only chance and chemical laws, but not natural selection, would be available to arrange amino acids.
Even then, with life absent, we have direct empirical evidence that amino acids do not spontaneously join together into proteins under natural conditions.
If outside of living things, amino acids don't polymerize into proteins, how can they do it inside living things?
It's not that the chemistry inside living things is magical and different. It's that life harnesses energy to operate in a state of chemical disequilibrium. Outside of life, chemical reactions go to equilibrium.
So hooking amino acids together to make proteins requires a state of chemical disequilibrium?
Yes, there are at least 4 things cells need to make proteins. First, a complex set of machinery to do the work. Second, a source of energy to drive the chemical reaction in a specific direction. That is the disequilibrium part. Third, information about how to put the protein together. And fourth, the amino acids that will be strung together. When organisms die, chemical reactions go to equilibrium. The protein-producing machinery disintegrates and proteins start to decay. They don't continue to be made.
In other words, nature outside of living things doesn't make proteins. If it doesn't make proteins at all, It can't make functional proteins by chance.
Outside of living things, the sophisticated laboratories of very intelligent chemists are the only known places where functional proteins might be made.
What if we assumed there was a natural law we don't know about yet that causes amino acids to join together into proteins?
Our present knowledge may be incomplete, but there is no empirical reason to suspect that there is some undiscovered law of abiotic protein construction. Even if such a law existed, the probability that amino acids would arrange themselves by chance into exactly the right sequences is unthinkable. Even given our huge universe and billions of years, probabilistic resources are still insufficient to create by chance even a single functional protein of reasonable size.
So, if chance doesn't get the job done, what about natural laws? If you have a universe with life-friendly natural laws, could they account for life?
Outside of life, natural chemical laws drive the disintegration of proteins, nucleic acids, and other large molecules essential for life. They don't cause their production. Natural law is sufficient to destroy proteins, but not sufficient to produce them abiotically.
So, natural processes are commonly observed to cause the death of living organisms, but have never been observed to cause life to arise abiotically.
By definition, laws do the same thing over and over again. Laws keep planets revolving around the sun and hold together the chemical bonds within biological molecules. But laws are not the right tools for joining amino acids together in long, complex, and highly specified sequences to make the thousands of distinct functional proteins found in organisms.
So, without life, there would be no proteins, and the same is probably true of other biological macromolecules. But what about once you have life? Could chance, coupled with the law of natural selection, account for some of the proteins found in organisms?
A protein called endothelin-1 will work well to illustrate why chance and natural selection are the wrong tools to make novel proteins, while design, if you're willing to allow it, makes sense. Inside our bodies, endothelin-1 acts like a key that fits into lock-like protein receptors that then signal for blood vessels to constrict.
Why is that important?
If all blood vessels were wide open, there would not be enough blood to fill them. Endothelin-1 works with other mechanisms to ensure blood doesn't just flow down into your legs when you stand up. By keeping many of the blood vessels in the lower part of your body and legs constricted, blood stays in the upper part of your body.
So, it prevents us from fainting from lack of blood to our brain each time we stand up.
Yes, but there's a lot more. Endothelin-1 also constricts blood vessels when we experience major trauma, like being shot or in a car accident.
Constricting blood vessels near wounds slows down bleeding, giving time for blood clotting to stop the flow before we bleed to death.
The importance of endothelin-1 should now be obvious, but there are other things in which it plays a role. For example, it helps to kill cancers by constricting blood vessels leading to tumors, thus starving them to death.
So endothelin-1 is designed to allow us to live normal lives without fainting every time we stand up, helps us survive injuries, and helps control cancer. It certainly is a powerful molecule.
It does have powerful effects, but remember that endothelin-1 is part of a much larger system. For example, if there were no receptor molecules for it to fit into like a key, endothelin-1 would be useless. Also, and this is very important, if it caused blood vessels to constrict, cutting off blood at the wrong place or at the wrong time, severe damage could result.
It would really be a problem if it cut off the blood supply to my brain.
Let's look at endothelin-1 to see if it might be the product of nature acting alone using only chance and laws.
If it is short and you have a random protein generator, then maybe endothelin-1 could be made by randomly joining together amino acids.
That's right. And endothelin-1 is in fact very short, only 21 amino acids long.
That is a very short protein. Most proteins are hundreds of amino acids long. It seems that a 21 amino acid long protein might just evolve by random combinations of amino acids.
To get the exact sequence of amino acids in endothelin 1 would be 1 chance in 20 raised to the 21st power, or about 1 chance in 2.1 octillion.
That would be 1 over 21 followed by 26 zeros. Perhaps it's not so likely.
A lot would depend on how big your random protein generator is. Also, it may be possible for variations on the sequence to still work, so that would increase the probability considerably. A Darwinist might argue that once you have living things, they act as random protein generators because of random mutations in the DNA that codes for proteins, and with lots of living things and a very long time, you could very well get a short protein like endothelin 1.
But don't most proteins start out longer and get processed into shorter proteins as part of their production process?
That's right. In the case of endothelin 1, it starts out as a 212 amino acid long protein called pre-pro-endothelin 1.
A 212 amino acid protein is probably too long to be made by chance, even with billions of years and millions upon millions of organisms experiencing mutations.
Probably. And the much longer pre-pro-endothelin 1 is full of important functional elements. For example, there are built-in signals for making the protein in the right location in the cell. Other signals act like postal zip codes to move the protein through the cell, package it up, and export it across the cell's outer membrane.
And there must be signals for removing these extra parts of the protein.
My favorite part is a safety catch that prevents endothelin 1 from causing blood vessels to constrict in the wrong place once it has been released from the cells that made it.
It looks as if the pre-pro-endothelin 1 protein has lots of information coded into it. But where does the information for making pre-pro-endothelin 1 come from?
That information is encoded in DNA in chromosomes. In the case of human endothelin 1, the information is encoded in DNA that is part of chromosome 6. This information is copied to RNA by the molecular equivalent of a DNA photocopier called RNA polymerase.
And then the information from the DNA that is now copied onto RNA is translated into endothelin 1 by the complex molecular machinery cells use to hook together amino acids. The parts of that machinery are made of both proteins and RNA themselves.
So information about the specific order in which amino acids must be hooked together to make endothelin 1 is encoded in DNA. But it is not just the sequence of amino acids that is encoded in the endothelin 1 gene on chromosome 6.
There would have to be special signals to tell cells when to make endothelin 1.
That's right. You don't want endothelin 1 in the wrong place or at the wrong time. Remember that it has powerful effects that could kill or injure you. One way cells control endothelin 1 is encoded into RNA copies of DNA-encoded information.
So production of RNA copies is controlled by signals encoded in DNA. When information for making endothelin 1 is copied onto RNA, part of that information includes instructions about how many endothelin 1 protein molecules to make using each RNA copy.
At the end of the endothelin 1 RNA is a signal that says to destroy the RNA after the cell's protein-making machinery has read it one time. So each RNA copy is read only once to make one endothelin 1 protein. If the information in the RNA copy was not destroyed, it could be used over and over again to make many endothelin 1 proteins, but that might be dangerous.
The RNA message is like self-destructing classified information. What is really amazing is how integrated the whole system is, and how much information is involved in making a tiny 21 amino acid long protein like endothelin 1.
The information for producing endothelin 1 stretches for about 8,000 nucleotide DNA letters.
There's obviously lots of information in DNA, but where does the information in DNA come from? Doesn't information come from the minds of authors, scientists, engineers, and other intelligent people?
Christians believe the information in living things ultimately came from the mind of God. For example, the Apostle John said,
"You are worthy, our Lord and God, to receive glory and honor and power, For you created all things, and by your will they were created and have their being.
Let's take a closer look at how information is encoded. Crystals show that nature can make repetitive patterns using laws. But the same thing repeated over and over again does not encode much information.
Yes, a crystal is like a 1,000-page book that only contains one short word like the or and, repeated hundreds of thousands of times.
Now that would be a boring book, because it contains little information.
Nature may also be able to use chance to make complex, non-repetitive patterns, but this would be like a book filled with random gibberish.
And that would be a boring book as well, as it would also contain little information. The exciting thing about life is that even the simplest cells contain large DNA libraries of highly complex and specified information.
Complex molecular machinery translates the DNA blueprint into protein machines. Remember, nature has two tools to work with: chance, which can produce complex but meaningless patterns, and laws, which can make repetitive patterns that also lack significant information.
On the other hand, information is routinely produced by intelligent beings. So, where information exists, intelligence was probably involved.
But doesn't the neo-Darwinian synthesis combine random mutations with the law of natural selection? That seems different from laws or chance acting alone.
There are a number of problems with even a combination of random DNA mutation and natural selection. First of all, natural selection must have a living, reproducing organism to work on. Before there were living things, natural selection could not act. So chance and chemical laws, but not natural selection, really is all nature had to work with if it alone made the first life, and chance is not a great way of making information.
Even with chance and natural selection coupled together, the selection can only be for making more offspring. It doesn't have any other goal in mind.
That is a profound problem for the Darwinian mechanism of chance DNA mutations coupled with the law of natural selection. It is not as if you can almost have the information to make a functional protein and expect natural selection to keep selecting until a functional protein is finally produced. Darwinian evolution has no goal it's aiming for.
Plant or animal breeders may select traits with some goal in mind, and even they can't create new kinds of organisms, just variations of what is already there.
Going from information for a protein to DNA coding information for a better version of the same protein would also be problematic unless each change along the way caused production of more offspring.
Changing information, even phrases with similar meanings, is very difficult without getting incoherent intermediates. With language, the meaning of intermediates becomes unintelligible. In the language of proteins, the protein ceases to function. In fact, when the information for some proteins becomes garbled by random mutations, organisms are frequently either killed or greatly handicapped. So natural selection can weed out organisms with mutated DNA. But if mutated DNA can't get in small incrementally improved steps to information for better proteins, natural selection can't help.
Darwinian natural selection, coupled with random DNA mutation, may be a real phenomenon in nature. But it does not account for the origin of either complex specified information encoded in DNA or the molecular machines the DNA codes for. Suggesting it does is like suggesting that because electromagnetism is real and printing presses run on electricity, electricity must write novels.
Neither laws nor chance, or even a combination of the two, accounts for the information in DNA. But we need to turn the same skeptical spotlight of logic and evidence on the possibility of design.
To claim that life is designed is to claim that it is the result of a decision made by an intelligent mind for a purpose. In some cases, the purpose may not be clear. Understanding purpose may require understanding the mind of the designer, but even where purpose is not understood, design may still be evident.
The design explanation is also the one favored by most Christians, including many scientists and philosophers.
Yes, unlike materialism, which for philosophical reasons denies the possibility of intelligent design when studying nature, the philosophical position of theism opens minds to the design option.
That philosophical orientation is probably why some critics, fairly or not, claim that there are no objective criteria for identifying design in nature, and it is just an arbitrary religious position.
That criticism isn't valid. Take, for example, the criteria used by archaeologists to determine whether a stone axe was designed rather than naturally weathered rock.
A number of criteria are commonly used to identify design. Recognizing design requires no specific religious commitment, only a mind open to the possibility.
Recently, 2 sophisticated identifying marks of design have been proposed: irreducible complexity and specified complexity. These features are thought to be reliable indicators of design, although they are not necessarily present in every object that has been designed.
Irreducible complexity refers to a system, like a mousetrap, where every element of the system is required in order for the system to function at all. If any part of a mousetrap is removed, it won't catch a mouse.
Such a system can't be assembled by natural selection one part at a time because, until the system is complete, it doesn't work. Michael Behe, who pointed out the connection between irreducible complexity and design in nature, defined it as
a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning.
The turbine-like protein machine called ATP synthase harnesses energy inside cells and is a biological example of irreducible complexity.
there is a gamma protein drive shaft between the FO subunit in the membrane and the F1 head where the ATP energy fuel of the cell is made. Without the drive shaft, the entire complex mechanism is essentially a boondoggle that does nothing at best and at worst may cause problems.
Natural selection won't create the other parts before the drive shaft because each component part has no purpose until the drive shaft is in place. And the drive shaft is also pointless without the other components.
ATP synthase, like many other molecular machines, does not reasonably look like the product of chance. It also does not look as if its components could have been shaped by natural selection before this fully functional molecular machine existed in the first place.
If it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive, slight modifications, my theory would absolutely break down.
So irreducibly complex molecular machines that could not evolve via numerous small steps show that Darwinian evolution fails at the molecular level. What about specified complexity? How does this idea expose problems with Darwin's theory of evolution and support the idea of design in nature?
Specified complexity describes things that are unlikely due to their complexity and yet fit patterns that they are not required to fit by some set of laws. For example, William Dembski has pointed out that a random sequence of letters may be improbable, but it is just gibberish. On the other hand, a sonnet by William Shakespeare is both complex and specified.
Shall I compare thee to a summer's day? Thou art more lovely and more temperate. Rough winds do shake the darling buds of May, and summer's lease hath all too short a date.
Yes, that is an excellent example of specified complexity, and we know that Shakespeare was a genius. If it took a genius to write Shakespeare's sonnets, then imagine what it must have taken to write the complex and specified information in DNA describing how to construct the poetry of a flower bud or the mind of a Shakespeare.
Chance is ineffective at creating complex and specified things, although it may create a complex mess. Information is created by intelligent minds, not by mindless physical processes.
William Dembski described how design can be recognized most of the time by using a conceptual filter which lays out the common sense thinking process rational people routinely use to differentiate design from laws and chance. It reflects the process already illustrated with endothelin 1. When determining if an object or event is intelligently designed, the first question is whether or not it is a product of natural laws.
That would be things that happen in the same way all the time, like hydrogen combining with oxygen to make water or gravity holding planets in their orbits.
If it's not a product of laws, then we ask if it might be the product of chance.
Like flipping a coin and getting 3 heads in a row. There's a fair chance of doing that, so if someone did it, getting the 3 heads would be reasonably attributed to chance.
Finally, we ask if it is complex, which means very unlikely, while at the same time also being specified. If it is both complex and specified, we conclude that it is intelligently designed.
Most people are familiar with probability and complexity, but specification seems more subtle.
The cylinders and pistons in an internal combustion engine are an example of specification. The iron from which these engine parts are formed may be molded into almost any conceivable shape, but they just happen to fit together in such a way that engines work.
There is no natural law that causes iron to form cylinders or pistons, or dictates that they should fit to a tolerance of several nanometers. They fit together because they were designed to do so by intelligent engineers with the goal of making an engine.
The information in DNA, coding for endothelin 1, is specified because it spells out exactly where, when, and how to make endothelin 1. Just as iron can be formed into any shape, the nucleotide letters of the DNA language can be arranged as gibberish or to encode any kind of information.
The DNA encoded information for making endothelin 1 is just right for making a protein that fits like a key into receptor molecules. That seems even more specified than the pistons and cylinders in an engine.
Since both chance and natural law are inadequate as explanations for endothelin 1 or ATP synthase, just as they don't explain the thousands of other proteins in a typical cell, they clearly do not explain how living things came to be. While natural laws and chance are out, design, supported by abundant specified complexity found in living things, remains a rational explanation for life.
Much of nature exhibits characteristics of intelligent design, whether informally stated as apparent purpose or more formally stated as specification and small probability. George Gaylord Simpson, one of the fathers of neo-Darwinism, wrote,
"This appearance of purposefulness is pervasive in nature, in the general structure of animals and plants, in the mechanisms of their various organs, and in the give and take of their relationships with each other. Accounting for this apparent purposefulness is a basic problem for any system of philosophy or of science."
Intelligent design explains the apparent purposefulness in life. The more nature is studied, the more marvels are revealed, and the more evident its brilliant design becomes. The Christian worldview, with its Creator God, allows a genuinely rational appreciation of the beauty in nature and the Creator who gave this gift to all living things.